In just the last few decades, biology has, well, it's just exploded. It's become this incredibly data-rich science. We're churning out information about the very code of life faster than we ever thought possible. But that's the thing, right? With all this data comes a huge, monumental challenge. How on earth do we make sense of it all?
I mean, really think about it for a second. This isn't just like organizing your bookshelf or even a giant city library. Nope. We're talking about trying to create a single unified digital catalog for every single gene, every protein, every tiny molecular machine that makes life tick. It's a massive global undertaking, to say the least.
And the sheer scale of it all, it's just staggering. Let's take just one database, okay? The Protein Data Bank. It holds over 200,000 unique, experimentally determined 3D structures, and that number's always growing. So you've got this data deluge, and that is exactly why we need a system, a kind of global digital library to manage this incredible wealth of information. All right, so where do we even begin? Let's start our journey of discovery right where most scientific quests do, with a single gene. So put yourself in our researcher's shoes for a minute. You're trying to understand a specific health condition. What's the very first question you're going to ask? Yep, which genes are involved? But it's almost never that simple. It's rarely a clean one-to-one relationship. The reality is way more like a complex dance between a whole bunch of genes and all sorts of environmental factors. So to even begin to untangle that web, scientists have to rely on these really specialized databases that are designed to map these very connections. And these are some of the heavy hitters. The first place any genetic detective is going to look. You've got databases like OMIM, which is like the super comprehensive catalog that links specific genes to known disorders. Then you've got others, like DisGenet, which casts a much wider net, combing through tons of scientific papers to find potential associations. The big takeaway here, these tools are what build that absolutely essential first bridge, connecting a little piece of genetic code to a real-world disease. Okay, great. So we've pinpointed a gene we're interested in. But remember, a gene is just the blueprint, right? It's the instructions. What about the actual machine it builds? That's the protein. So what's the next logical question? Of course, what do these little molecular machines actually look like in 3 dimensions? And that is exactly where primary structure databases come into play. Think of them as the foundational archives, the vaults. They store the raw, experimentally determined 3D coordinates of these molecules. It's like the ultimate source of truth for a molecule's shape. No interpretation, just the raw data. And the undisputed king of these databases, that's the PDB, the Protein Data Bank. I mean, this thing was established way back in 1971. It is the single global repository for all of these structures. If a scientist anywhere in the world figures out a new protein structure, this is where it goes, so the entire world can access it. It's just an incredible, incredible collaborative resource. But here's the thing, just having hundreds of thousands of raw structures isn't quite enough. I mean, what do you do with all that? We need a way to sort through them, to find the patterns. And that is the job of secondary or derived databases. They add this crucial layer of analysis and classification right on top of the raw data from the PDB. You could say they're kind of like the librarians of the molecular world. And this really highlights a key point. There's more than one way to organize this library, right? For example, SCOPE and CATH are 2 of the big classification systems, and they take pretty different approaches. You can think of SCOPE as the meticulous human expert, carefully curating everything based on evolutionary relationships. CATH, on the other hand, leans more into automation, classifying structures based on their architecture and topology. And here's the cool part. Both are totally invaluable. They just give us different, but equally important ways of looking at the very same data. All right, let's recap. We know our gene, and we've got a good idea of what its protein looks like. But a gene isn't always on. It isn't always active. So the next really crucial question for our investigation becomes, in which cells and under what specific conditions is this gene actually switched on? We call this gene expression. And let me tell you, understanding this is absolutely critical. It's not just a nice to know. As this quote really drives home, knowing when and where genes are expressed can actually help us predict the course of diseases like cancer. It helps us uncover the fundamental biology of how our cells even work. So how do scientists actually get this data? Well, it's basically a 3-step process. First, they gather a sample, cells from a specific tissue or condition. Second, they use these really powerful techniques, like microarrays, which are amazing. They act like a genetic switchboard that lights up to show you exactly which genes are active to measure thousands of genes all at once. And third, and this is the most critical part, they upload all that well-documented data to public repositories like GEO, making it available so the entire research community can benefit and learn from that one experiment. Okay, so we're really starting to build a detailed picture here, aren't we? We've got the gene, we have its protein structure, and we even know when and where it's active. But here's the thing. No gene or protein ever works in a vacuum. So now, let's zoom out. Let's see how our little piece of the puzzle fits into the vast, interconnected network of the entire cell. Because really, the old saying, no man is an island, is true for molecules too. To understand its true function, you have to place it in its proper context. And that context is what we call a biological pathway. You can think of these as the roadmaps of the cell, or maybe even the wiring diagrams that show exactly how all the different molecules work together to get stuff done.
So one major type of pathway is all about signaling. This is basically the cell's internal communication system. A signal comes in from the outside, and it kicks off this whole cascade of interactions that passes the message along, kind of like a relay race, to create a specific response.
A second major type is gene regulation. Now, these pathways are the ones that directly control which genes get turned on or off. They're what link the signals from the outside world to the cell's genetic blueprint. And you see? This connects right back to what we were just talking about with gene expression.
And then, the third type you'll see are metabolic pathways. These are the assembly lines of the cell, the chemical factories, and the recycling plants. It's how the cell builds things, breaks things down, and generates energy. And there are incredible databases like KEGG that are dedicated to mapping these unbelievably complex networks of reactions that, well, keep us alive. So look at how far we've come, from a single gene to its protein to its activity, all the way out to the entire cellular network. It's a pretty amazing journey of discovery, and it's all made possible by this incredible global data infrastructure. But that leads to the big question. What does the future hold for this constantly expanding universe of biological information?
Well, using this massive digital library effectively means you have to be a bit of a savvy consumer. The data isn't always perfect or complete. A good researcher knows that. They know that information might be missing, so they have to check multiple sources. They have to watch out for potential errors or data that's become redundant. In the end, it's all about integrating information from lots of different places and doing it intelligently. But look, despite those challenges, the main takeaway here is crystal clear. These biological databases aren't just helpful, they are an absolutely invaluable, indispensable foundation for pretty much all of modern biological research. They make everything we've just talked about even possible. And that leaves us with one last and I think really exciting thought. We as a global scientific community have built this incredible, interconnected digital library of life itself. And it's at the fingertips of anyone, anywhere with an internet connection. So the only real question left to ask is, what amazing discoveries are we going to make with it next?